Hello, good afternoon. My name is Monica Ponder, and I'm an assistant professor in the Department of Food Science and Technology. And I believe I am your representative of your entry into the interest in nanotechnology and food sciences. Mine is not necessarily the focus on how to use nanotechnology as a food preservative method, but that is definitely one way that we could do it. I am a microbial ecologist with an emphasis on understanding how pathogens and normal microbiota interact to affect the host. Ultimately, I'm trying to improve gut health. And so my interest in this nanobio began out of a collaboration that one of my collaborators in the department, Dr. Sue Duncan, had with Andrea Dietrich, who is in the nanobio surface water thrust. And I think that's my next slide. Dr. Dietrich has been interested in trying to understand how you can improve the palatability of iron compounds for cancer patients. She and Dr. Duncan have been working in that area. And through attending the lectures of their students, I began to get very interested in what do we really know about these nanoparticles. We don't really understand how the bioavailability of iron is affected by having it as a very small compound. So is it more readily available to the host itself? And is it also readily available to the other microorganisms within that host? Nanoiron is being used commercially. It's being used for removing contaminants from drinking water, and you can actually detect appreciable amounts of nanoiron in normal municipal water supplies who are using these treatment sources. And there's also a number of different nanoiron particles that are being considered for addition to meal supplement bars. These are bars that we provide as food aid to developing countries in times of famine. And so asking questions about whether or not this nanoiron can become more available for potentially pathogenic bacteria is a really important consideration when you start looking at those vulnerable types of populations. Also, eventually, we would like to be able to combine these nanoparticles with things like probiotics and prebiotics so that you could target and stimulate the growth of particularly beneficial types of bacteria within a host, and that therefore you would promote some type of health promoting, therefore making a functional nanoparticle as part of your ingredients. So this is really our first foray into this type of research within the Department of Food Science and Technology, but we're definitely interested in continuing it further. So what we have done in just the couple of months that we've had this funding is trying to understand what the effect of the nanoiron in all of its valence scales, either a nanoviolet or different iron oxides, are on the human enteric pathogens. So we took a panel of some of the most commonly occurring human enteric pathogens. So these would be Salmonella enterica, E. coli, Vibrio, some Vibrio, and Listeria monocytogenes. And we simply added nanoiron at a comparable composition as to what you would see in a drinking water or in some of the proposed meal replacement items, and to see how that influenced the growth and the survival of those pathogens that we are looking at. And we found that it actually was able to improve the growth of Salmonella enterica. There was no effect on two of the other pathogens that we looked at. But what is particularly troubling is that Salmonella enterica is endemic, and it causes very profound and profuse diarrhea in vulnerable populations. So this suggests that we need to continue to understand ways to deliver this nanoiron. One of the things that Dr. Dietrich and I will be doing in the future is looking at different surface coatings of that nanoiron to try to make it more bioavailable to the human host or to the animal host. We're interested in moving into a mouse or into a porcine model, and less bioavailable to the microorganisms that we could be using. So we should be hopefully moving into some coating strategies, encapsulation, and also comparing different commercial nanoparticles that are out there that have different coatings associated with them to see how that influences either the bioavailability to the human host or to the pathogen itself. And we're also going to be examining how the overall microbial community is influenced by the presence of nanoiron in there. We would like to see stimulation of some good bacteria, things that are going to be synthesizing butyrate and other things that are known to improve the health of the organism itself. And we plan on pursuing some federal funding for the next round, targeting right now the National Institute of Environmental Health and Sciences. They have an R21 that's going to be looking at the environmental influences on the microbiome with using these nanoparticles as being bioavailable within the environment, and also targeting USDA AFRI with their food biotechnology.
I was kind of surprised that iron didn't enhance the growth of listeria. You know, when pigs are given parental supplement with iron, they come down with Listeria monocytogenes as well as E. coli